Before I move on, I want you to realize how important it is to remember from your text and from the supplemental videos I provide to you this week that cancer is not a single disease and it does not arise from a single hit. A normal cell sustains many insults or exposure to damaging agents, chemicals, radiation, viruses, etc. And most of the time these insults cause DNA damage. And most of the time the cell responds in an appropriate manner, either repairing the damage, going into senescence, or apoptosis. It's when the cell receives too much damage that the protective or corrective mechanisms are overwhelmed, that we start to see activation of oncogenes or loss of tumor suppressor genes or loss of apoptosis, and then malignant neoplasms arise. So it is all about initiation of DNA damage by some physical agent that overwhelms a cell's corrective mechanisms. DNA damage leads to DNA mutation, a fixed, heritable change, meaning it is now fixed in place in the genetic code, and this change can be transmitted to future daughter cells. There is further genetic insult in the activation of oncogenes, loss of tumor suppressor genes, etc. in the promotion phase, and then a neoplasm that is able to recruit a blood supply, has uncontrolled growth, invasiveness, it goes through the basement membrane, lacks differentiation, etc. And this occurs in the progression stage of cancer. Now, how does a proto-oncogene get activated or mutated into an oncogene? There are several different types of DNA mutations: a point mutation, translocation, or even a duplication/amplification of a gene. Even insertion of viral DNA that leads to increased expression of a proto-oncogene. You might be wondering how a gene could be duplicated or amplified. Well, that's usually from viral infection. Some viruses insert their DNA into our genome, and when their packaging machinery clips their DNA back out of our DNA, sometimes they pick up a gene from our genome, and this can be reinserted into another cell, and now that other cell would have an extra copy of that gene. An example of an amplified gene that leads to cancerous growth is the human epidermal growth factor receptor 2, or HER2. This gene is amplified in about 30% of breast cancers, and its overexpression leads to a poor prognosis due to the aggressive nature of this neoplasm. An agent used to treat this cancer is Herceptin, and it is a monoclonal antibody, which I will discuss more later in this lecture. The antibody binds to the receptor and renders it inactive. Therefore, the neoplasm can no longer respond to the epidermal growth factor because its receptor is blocked by the antibody. A classic example for a point mutation generating an oncogene is the story behind the RAS oncogene. It's really not just one oncogene, it's a family of oncogenes. These RAS proto-oncogenes normally act as second messenger systems that send growth signals to the nucleus to increase cell mitotic rate. When a point mutation occurs in these genes, it leads to a gain of function, and now they are oncogenes that promote too much cellular division. What about translocations? Well, some chromosomal ends are more, sticky than others, or are more prone to double-strand breaks, or DSBs. As such, you can have two chromosomes trying to seal their ends by binding with each other. If it's a reciprocal translocation, a person may be lucky in that they still have the normal number of chromosomes in their cells, and there is no disruption of genes where the chromosomes split and reunited. But sometimes this reciprocal translocation can disrupt a gene, or maybe even create a fused gene in the splice site, where the gene is now in close proximity to a different promoter that leads to excessive expression of the gene. Now I'd like to tell you about the translocation that occurs to form the Philadelphia chromosome. In most cases of chronic myelogenous leukemia, as well as some cases of acute lymphocytic and myelogenous leukemia, a piece of chromosome 9 and a piece of chromosome 22 break off and switch places with each other. This type of damage is called translocation. This new chromosome, which is mostly chromosome 22 with a piece of chromosome 9 stuck to it, is called the Philadelphia chromosome, designated here at the right of the picture as PH superscript 1. So now chromosome 9 gets longer and chromosome 22 gets shorter due to this swapping of DNA between the two chromosomes. The Philadelphia chromosome is only found in the affected blood cells. Because of the damage to the DNA, the Philadelphia chromosome results in the production of an abnormal enzyme called tyrosine kinase. Tyrosine kinase is a huge player in one type of second messenger cascades and sends growth signals to the nucleus. Along with other abnormalities, this enzyme causes the cancer cell to grow uncontrollably. When one change in the DNA occurs, this leads to further genomic instability. Again, it's not just a single change to the DNA, but rather a cascade of genomic events that lead to uncontrolled cell growth. So, just to review, proto-oncogenes code for proteins that promote cell growth and division. They can be growth factors, the example here being IGF or insulin-like growth factor. Receptors for growth factors like HER2, or signal transduction factors like cytoplasmic kinases, the example from the previous slide being tyrosine kinase. The ultimate target is going to be DNA and transcription of genes into proteins, transcription and translation, that are involved in the cell cycle, shown here as cyclin D. So, shown more graphically here, a mutation in a proto-oncogene leads to a gain of function, and this oncogene can code for more increased growth factor, picture 1, more growth receptors, picture 2, increased second messenger system activity, picture 3, and all of this leads to an increase in gene transcription or expression of genes that code for cell cycle proteins like cyclins, shown in picture 4. Let's switch now to tumor suppressor genes. Tumor suppressor genes code for proteins that inhibit cell growth and division. They allow for the timing of our cell cycle checkpoints. The classic examples are p53 and RB. A picture of both is seen here. The gene for p53 is sometimes called TP53, or we use italics when we write p53 when we speak of the gene. We can discuss either one, and the change in the genetics is similar, so I'll start with p53. It is a protein that halts the cell cycle and allows for DNA damage assessment prior to DNA replication during the S phase. Looking at the bottom picture, we see a normal p53 arresting the cell cycle and allowing DNA repair enzymes to repair DNA damage. If there is too much damage, we see p53 inducing apoptosis. Now, how can we get an abnormal p53 or, for that matter, RB protein product? Turns out these genes use typical Mendelian genetics. By that I mean you can be homozygous for two normal copies and you would have normal p53 or RB proteins. You can also be heterozygous for a normal p53 or RB gene, and you would still make enough of those proteins to regulate the cell cycle. However, if you lose your heterozygosity, or LOH, and now have two mutated copies of these genes, you now fail to make p53 or RB, and now you lose your checkpoints. In other words, you need a two-hit mechanism before these systems fail. The first damaged copy can be inherited, as shown at the top picture, demonstrating inherited retinoblastoma, versus sporadic retinoblastoma, where both copies of the RB gene are mutated by some agent at different times. The end result is if a cell lacks these proteins, then they fail to stop cell division and allow for DNA repair. The cell continues to divide without repair to damaged DNA, and the damaged cells continue to divide. The genome becomes progressively unstable, and eventually the cell turns cancerous. Viral oncogenes are responsible for oncogenesis resulting from persistent virus infection. Although different human tumor viruses express different viral oncogenes and induce different tumors, their oncoproteins often target similar sets of cellular tumor suppressors or signal pathways to immortalize and/or transform infected cells. In 1909, a farmer brought Dr. Francis Peyton Rouse, a junior faculty member then at Rockefeller University, a hen that had a breast tumor. Rouse performed an autopsy, extracted tumor cells, and injected the cells into other hens, which then developed sarcoma. This was the first experimental proof of an infectious etiologic agent of cancer, and the chicken sarcoma-inducing RNA virus was subsequently named the Rouse sarcoma virus. After a half-century debate on whether viruses truly cause cancer, Rouse was eventually awarded the Nobel Prize in Medicine and Physiology in 1966 for his discovery of tumor-inducing viruses. It is now estimated that 20 to 25% of human cancers worldwide have a known viral etiology. Early pioneering efforts on tumor-inducing viruses were mainly focused on avian and small animal retroviruses, a group of RNA viruses containing an RNA-dependent DNA polymerase, or reverse transcriptase. And it was thought that there were no similar viruses in humans. Demonstration in 1980 of the first human retrovirus, human T-cell leukemia virus type 1, or HTLV1, which causes adult T-cell leukemia, was therefore a landmark achievement. Viral strategies for manipulating the expression of cellular genes to enhance viral persistence, viral latency, and survival of infected cells have provided numerous clues to the mechanisms of gene expression and their dysregulation during tumor development. The fact that all characterized viral oncoproteins are sticky and interact with cellular proteins in infected cells makes each of them multifunctional and essential for cell immortalization and transformation. By interacting with several dozens or even hundreds of cellular factors, viral oncoproteins from different tumor viruses eventually end up in apparently similar scenarios of viral carcinogenesis by targeting cellular tumor suppressors, deregulating signal transduction pathways, redirecting gene transcription, and or stimulating anti-apoptotic programs in the host cell. There appears to be an epigenomic component to the development of cancer. In this hypothesis, there isn't a mutation that is occurring to the gene. Rather, external environmental influences cause changes in gene expression. In this picture, you are seeing chromatin, and chromatin is DNA wound up around histone proteins. The DNA must be unwound from these proteins for gene expression to occur. External environmental influences, drugs, aging, development in utero, diet, etc., can cause changes in the histones and their methylation status, shown here as a change in a, quote, histone tail. If we apply this concept to our previous slides, it's not hard to understand how a neoplasm could generate through changes in methylation of histones that lead to increased gene expression of proto-oncogenes or decreases in expression of tumor suppressor genes. Patterns of gene expression can be altered in one individual And epigenetic studies now suggest that this change in gene expression could be transmitted to future generations. If we go back to our previous lectures, we are reminded of the Holland hunger winter and how subsequent generations have had more problems with obesity, cardiovascular disease, etc. Well, some studies show, and I'm just throwing out an example that I think I remember reading about, that if a man had a grandfather who smoked, that man was more likely to develop, I think it was testicular cancer. The other contributing hypothesis for neoplasm development is failure of the immune system to remove cells that express tumor-associated antigens. When a cell develops into a neoplasm, we recognize that there is altered gene expression and genomic instability. Part of the altered gene expression is the expression of different proteins that are inserted into the cell membrane. These new antigens are new antigens. These are tumor antigens and should be detected by your cytotoxic T cells as foreign and the tumor destroyed. However, the neoplastic cells, with their unique gene expression, are able to escape detection and avoid destruction by the immune system. There are some experiments and data to support this contributing hypothesis. There is a higher incidence of neoplasia in the immunodeficient and immunosuppressed. As we age, our immune system becomes compromised, and there is a correlation between age and incidence of cancer. That meaning the older we get, the more likely we develop cancer. But there are also challenges to this hypothesis. For example, patients who have a thymectomy show no increase in neoplasias compared to their counterparts that have their thymus. With the expression of different antigens, or even different amounts of proteins in neoplastic cells, we can detect these proteins and use them as diagnostic tools or markers for different types of neoplasms. Neoplasms, as already discussed, are poorly differentiated and even revert back to expression of fetal proteins, and we see this expression of fetal antigens in some types of liver cancer. In some cases, aberrant hormone production is seen in some cancers as the cells become de-differentiated. And you can read the list of this the rest of this list if you want. I think you get the idea. Of course, another cause of cancer can be due to overexposure to a carcinogen. A carcinogen is an agent that is known to cause cancer or increase the incidence of cancer in animals or humans. For example, chimney sweeps were usually young boys or men that would climb in out of chimneys, and the soot had carcinogenic properties, and therefore there was a higher incidence of testicular cancer in these young men. I want to reiterate, however, that most cancers have a multifactorial origin. By that I mean you have to have familial predisposition to cancer, maybe through epigenetic changes in gene expression or inherited mutations. You need to have chronic exposure to a known carcinogen, etc. And there are many yet to be identified environmental agents that are likely to contribute to cancers. And this is probably the case for the great majority of cancers, because 95% of the time we don't know the exact etiology of the cancer. How do carcinogens work? They can be proximate or direct acting. This means they act locally and don't require some sort of metabolic process. Or they can be indirect acting, which means it needs further metabolic process before it becomes activated into a compound that can cause neoplasm. This type of agent is considered to be a pro-carcinogen. When considering a direct or indirect agent, they both ultimately cause some type of reactive oxygen species, a type of which is a free radical. Free radical oxygen species can cause damage to cellular macromolecules like DNA, which can lead to excessive DNA damage that overwhelms the DNA machinery and leads to mutations. And, well, there we go with the aberrant gene expression of proto-oncogenes and tumor suppressors, or failure of apoptosis all over again. The effects of carcinogens are usually dose-dependent. This can be a combination of the amount you are exposed to, as well as the duration you were exposed to that agent. Now, I want to tell you something interesting that we discovered while I was a graduate student. I worked in a lab that studied DNA damage and repair. We treated transgenic mice that overexpressed DNA repair genes with different carcinogens, with the idea that they would be able to withstand greater doses and longer exposure times. We used methyl nitrosourea, or MNU. We treated transgenic mice and normal mice with this agent. Both groups received either a high acute dose or a lower chronic dose. And we saw an interesting phenomenon in both mice. When treated with an acute, very high dose of MNU, we actually saw higher DNA repair in both sets of mice and fewer tumors than mice injected with a placebo. And we would joke and say that we should go and inject or expose ourselves to carcinogens in a one-time acute high-dose fashion to actually reduce our chances of cancer. So keep that in mind. Just because you're exposed to a carcinogen once doesn't necessarily mean that you will develop cancer. Chronic, low-dose exposure seems to have more deleterious effects. So if your patient has cancer, what would you expect to see? What are some of the manifestations? First, we know a neoplasm that has metastasized is an aggressive neoplasm that has the ability to enter the bloodstream and take up residence somewhere else in the body and recruit blood flow to deliver nutrients and oxygen. These are very greedy cells. As such, your patient will present with wasting syndrome. Anorexia refers to reduced food intake resulting in weight loss. The person is not ingesting enough nutrients even for their normal cells. People with cancer oftentimes have a decreased appetite. But anorexia alone in cancer patients does not explain the profound wasting syndrome we see in the patients. Cachexia means the normal cells are starving because even if the person were to eat or get nutrients, the cancer cells are taking more than their share of the nutrients. In other words, the metabolic demands of both the cancer cells and normal cells are not being met by the nutrient intake. The cancer cells especially are in a hypermetabolic state. There is even altered metabolism of the nutrients by the cancer cells, and all of these factors lead to significant weight loss. Lack of appropriate glucose for other tissues of the body, like the brain, can explain the appearance of sleep disorders, which leads to extreme fatigue in the patient. Lack of nutrients for immune cells can mean the patient becomes more susceptible to secondary infections. The tumor itself can cause organ failure in the primary site or even in secondary sites. Lack of nutrients for normal blood cell synthesis can lead to anemia, or the invasive nature of the cancer can cause hemorrhage outright. For example, colorectal cancer, the patients have blood in their stool very often. And other syndromes resulting from the initial cancer, called paraneoplastic syndromes, can arise. This basically means the cancer may cause, or act, I should say, as if it were an endocrine gland. For example, maybe it can release copious amounts of hormone, and now the person will present with another syndrome from the aberrant quantities of that hormone. For example, some lung cancers produce ACTH, and this can lead to huge quantities of glucocorticoid production in the body, and the person would present with Cushing's syndrome. Grading a tumor is a matter of assessing the degree of differentiation of a tumor, meaning what do they look like? Do they still resemble the parent tissue, or have they morphed into very pleomorphic, many more form cells? What is the mitotic index? Is it high due to high proliferation, etc.? And we use words like well differentiated or poorly differentiated. With staging, we use the TNM classification. T stands for tumor, N stands for nodes, and M stands for metastasis. For the T values, the representation of the T values represent the extent of the tumor spread, with the best designation being T0 for no evidence of primary tumor, like if the tumor recessed, to the point of no detection. TIS means carcinoma in situ, and this is a common diagnosis for breast cancer, a certain type, like ductal carcinoma in situ. You get the idea. A similar nomenclature exists for the number of lymph nodes that the cancer has metastasized to, and again, the lower the number, the better. And then lastly, the metastasis nomenclature to address other sites of metastases other than lymph nodes. Of course, the go-to for cancer treatment has long been and still is the surgical removal of the cancer. Obviously, some neoplasms are easier to get to than others, so this might not always be an option. Also, removing as much of the tumor as possible means a better prognosis. I was reading a paper last year that talked about the difficulty in removing brain tumors because obviously the surgeon would like to remove all of the tumor, but not remove healthy and much needed neurons. So it turns out the scorpion venom binds to cancer cells, and when you shine a black light on it, the cells fluoresce. So now the surgeon can see which cells are the cancer cells and leave behind the healthy neurons. Pretty cool, eh? Radiation therapy can be used. These are high energy waves that disrupt the cancer growth. However, there needs to be some access to the radiation therapy, and not all neoplasms lend themselves to radiation therapy, or they might be resistant to it, for example, melanomas. The radiation waves induce so much DNA damage that the cancer cells cannot continue to replicate and divide. Radiation therapy can be used alone or as an adjuvant, which means along with some other type of therapy, for example, surgery or something. There are different types of chemotherapy, and your book lists many of them. The idea is the more selective the chemotherapy in targeting the cancer cells, the fewer side effects the patient will experience. Chemotherapy targets rapidly dividing cells, and this is the reason why hair follicular cells end up being targeted by the chemotherapeutic agent and the patient loses their hair. Different chemotherapeutic agents have different receptors, and if the cancer lacks a receptor, then the chemotherapy is ineffective, if it lacks the receptor for that drug, of course. This is part of the problem with treating, for example, triple-negative breast cancer, because 3 receptors that could be targeted are not made by this cancer, and so now the patient has fewer chemotherapeutic agents to use against the cancer. These chemotherapeutic drugs can be direct-acting or indirect-acting, and will basically impair the cancer cells from dividing. Interestingly, when a woman has an ectopic pregnancy, that happens in the abdomen, not uterine tubes, and the developing embryo cannot be found to remove it, chemotherapy can be given to that woman, and the agent targets the rapidly dividing cells of the embryo to destroy it. My cousin had this type of ectopic pregnancy, and her family could not understand why they were treating her with anti-cancer drugs. They live in a very rural area of the country and have very little to no college education. Immunotherapy can be non-specific agents such as the Bacille-Camet-Guérin, it's French, or BCG. This is a strain of bacterium that causes bovine tuberculosis, and it non-specifically boosters the immune system and can help with treating, like bladder cancer. Some types of immunotherapy include taking matched natural killer cells or T lymphocytes from people who have low incidence of cancer in their families and then they give them to people with tumors, like a blood transfusion. The idea being that these immune cells would attack the tumor cells. So many more examples are being used, but let's move on. Biological modifiers include cytokines, or interferons, monoclonal antibodies, and blood growth factors. Interferons are proteins made by cells in response to a variety of cellular or viral stimuli. There are three major types, alpha, beta, and gamma, each group representing the receptors they bind to on their target cells. Interferons inhibit viral synthesis and tumor protein synthesis, and thus more cancer cells pushed into G0. Interferons can also stimulate the immune system, which, as previously noted, can help eradicate the tumor. Monoclonal antibodies are made by injecting a mouse with a cancer antigen. The mouse starts to make antibodies to that cancer antigen. We then sacrifice the mouse and harvest the immune cells from the spleen that are making antibodies. Then we fuse the mouse spleen cells with tumor cells, and the tumor cells help the spleen cells to continue to replicate in vitro. This is called transformation. This creates a hybrid cell culture called a hybridoma. You then plate this hybridoma cell line so that only one cell gets into a dish, and then you spend weeks, months, years trying to find the one hybridoma cell line that is making the antibody against your cancer antigen. Now stop and think, why does it take so long? Well, of course, there are many B cells in the spleen when you harvested it. Each B cell makes its own antibody. So you create thousands of hybridomas from that one spleen, but only one of them will make the antibody to the cancer antigen you initially injected into the mouse. These antibodies then can target the tumor cell in the patient and inactivate it in different ways. The picture on the bottom shows how monoclonal antibodies can be used to deliver drugs to a cancer cell, like dropping a bomb of radioactive substances or chemotherapeutic agents. And this basically means the antibody is conjugated to that drug. Obviously, this would mean specific targeting of the therapeutic agent to the tumor cell, and this would mean potentially fewer side effects. Or you could see on this picture that the monoclonal antibodies could even guide killer T cells to the cancer cells, just to point out another example of their uses. Hormone therapy can include delivering an agent that binds to a hormone receptor. Some cancers are supercharged from hormones, and thus blocking a hormone's receptor can block how rapidly the cancer grows. Tamoxifen works in this capacity by blocking the estrogen receptor and is used to treat some types of breast cancers. Well, this concludes our discussion on Chapter 8, Neoplasms. I hope you enjoyed what you heard, and I hope you've learned something.